So it's my pleasure to introduce today's fourth and final speaker, which is Khar. So a little bit of background on Karen. She got her PhD from the University of Kansas there in their Center for Computational Biology, where her studies were focused on using Rosetta molecular modeling to model dynamics of proteins, specifically looking at how proteins bind small molecules. And she's currently a scientist at Cyrus Biotechnology. And so we're going to hear about some of her work today from Cyrus Biotechnology, where she is a protein modeling scientist.
Excellent. Thank you for inviting me. I'm excited to talk a little bit. So this is a very different talk than the last three because I'm focusing on the protein modeling, the protein design parts of it. So as Allison mentioned, my background, I got my PhD in a Rosetta lab, so I learned how to use Rosetta to do some very specific protocols for protein modeling. But today I'm going to be describing a enzyme design method example using the Rosetta molecular modeling suite. So I'm showing an example instead of some actual data that we've done because we're a small biotech and we're not allowed to share any of the things we've done for people or with people. So I've just used this example of taking alpha amylase, and I'm going through the protocol of how I would generate a library of stabilizing mutations for testing in the wet lab. And of course, enzyme stabilization is super useful for a lot of different goals. In the last talk, they were talking about the need to stabilize the enzyme so that it can express a lot more. That's a pretty common thing that comes up. A lot of people need to do a lot of manufacturing and storage, so that's a key component that stabilization can help. But then people who are working on making designs in the active site of an enzyme to change the substrate specificity or change the activity of the enzyme, a lot of times it's extremely useful to start with just finding some stabilizing mutations for the enzyme. So then you have a good stable enzyme so that if you're creating active site mutations that are very helpful but turn out to destabilize in the protein, it makes your enzyme much more compatible with those sort of things. So you can make much more aggressive possible mutations if you start with a stabilized enzyme. So we're using the Rosetta molecular modeling suite, and I've been in the Rosetta community for about 11 or 12 years now. So Rosetta is a very wide range of suite of data or libraries that can help you model for a lot of different functions. So some people do GPCRs or membrane proteins or antibodies, and there's definitely several enzyme labs in the Rosetta community. But the cool thing about Rosetta is it's got so many codes you can mix and match to really tweak to your specific goal. But since it's so broad, it's pretty hard to learn a specific function. So one of the things my company has done is created some software that took the most commonly used protocols and it created an online GUI. So it's very push button. So I'm going to show you an enzyme sample using this online GUI here, which is called Cyrus Bench. So this is just an overview. I'm going to talk about Rosetta a little bit and then specific Rosetta tools in Bench that are used for stabilization or other enzyme modeling goals. And then I'm just going to show in this example some enzyme structure preparation, either optimization of a crystal structure or structure prediction if you don't have a crystal structure. And then I'm going to talk about the actual mutational sampling. And I'm going to specifically talk about finding single point mutations with scanning, site saturation mutagenesis, or a combination method to like choose a region of your protein and sample a bunch of mutations to find combinations that stabilize. Okay, so moving on, first off, Rosetta. Rosetta's been around for 20 plus years. It's got a lot of great code for protein modeling and one of the key features is the Rosetta enzyme, the Rosetta energy score term. So the Rosetta score terms, there's a lot of variations you can use, but the default version looks at all the atoms in your protein, looks at the bond lengths and the bond angles and how close each atom is to each other, and makes physics-based score on a conformation. And it also uses statistics-based things like a side chain conformation. There are certain conformations that are most prevalent in the PDB. So how similar are your conformations, different those things found in the PDB? So all these things combined will get one score term. And so these are incredibly useful in defining whether a mutation is good or a conformational change is good. So and the score term is updated quite regularly based on new experimental information. There are a lot of other things you can do to analyze a structure in Rosetta. Most of these here are things that we can do on a fee-for-service basis, and I'm not in the default bench GUI, but because there's just so much in Rosetta, we can't put everything in the handy GUI. Another thing that's a real powerhouse behind Rosetta is the use of the Monte Carlo simulation. So whenever it's sampling conformations or sampling mutations, it goes through this step. For stage one, you go through some sort of change, and that could be just sliding their things through their range of motion a little bit, or it could try some big jumps. So then it uses the Rosetta integrating function to score that change, and then it uses the Metropolis criteria to decide if it will accept that change. If the score gets better, it always accepts it. But if the score is unfavorable, it could still occasionally accept it if the score isn't too bad and you've got the temperature up high. So this is really key because it allows you to get out of those energetic hurdles. So you can really jump around in the search space and sample some things over here and then sample some things over here. So this is one of the key things that allows Rosetta to basically cheat, and compared to things like molecular dynamics that really has to slide through like physics-based movements. So a lot of the protocols use Monte Carlo. So next, I just want to look at the concept of optimization of a crystal structure. So when you're going to do design, you want to have like a high resolution crystal structure in the best case scenario. But even though you've got a really good crystal structure, you'll still want to optimize that structure a little bit, because a crystal is not what the normal native conformation of your protein is 100%, because you've got crystal packing artifacts, or whatever the crystal conditions were, it might shift things around a little bit. So one of the first things we like to do with a crystal structure is run the relax protocol. So relax is just a great way to take your protein, wiggle it around a bit using this Monte Carlo approach, and find a nearby energetic minimum. So here's just an example showing two crystal structures of the same protein, one in blue cartoon, one in red cartoon. But if you start with the blue cartoon and just run a relax, you'll find that you can optimize the structure, and you can end up in both of those minimums with depending on how many trajectories you run. So it's able to easily find nearby other favorable conformations in addition to relieving those crystal artifacts. So relax is one of the first protocols. And I'm going to jump back and forth a little bit between the bench software to show how we do these protocols in the bench software. So the software is just found online at cad.cyrusbio.com. So this is the interface, and so we load structures, and anytime we load a structure or create a new structure, they'll appear over here on the left. So I loaded this crystal structure 5U3A, and that structure is shown here in the structure viewer. And I've just highlighted key residues in this enzyme. So the catalytic residues are deep into this pocket right here. So the pocket that's important for binding specificity are highlighted here. And other key features would be these residues that are important for the calcium binding. So I've loaded this protein, and I'm looking to do enzyme stabilization. And so in order to stabilize it, I'm looking for regions where I can find improved mutations that aren't going to affect the active site. So one of the key points is I want to avoid making mutations directly in the active site, so that I can find stabilization that is not likely to affect the activity. I don't want to disrupt the binding of the substrate or the catalytic sites or the calcium binding. So that leaves a lot of potential protein space here, where if I were to find stabilizing mutations, it's unlikely to affect the active site function. And of course, that's 100% not guaranteed. So there's still a trial and error approach where you try designing in different parts of the protein to see how it affects the active site. But let's see, I'm just showing that I loaded this protein here, and Rosetta scored the protein looking at all the atoms and the bond links. And so the total score is -525, which is kind of meaningless on its own, but it's this base score that it's going to be using to try to optimize to lower that score by conformational changes or mutational changes. So I took this structure and I select an action to optimize the structure. So I just want to click the relax option and it opens up the screen. And so I want to do maybe 50 repeats of relax and click save and run. And so that's gonna run pretty quickly, but probably not done while we're talking. So I'll show you what the output of that would look like. So here's the output where it listed all 50 structures that ran through this relax protocol, where it just did this wiggling and find a more energetically favorable conformation. And so this is a Monte Carlo. Each has its own trajectory and it's a random set of changes. So they're all coming out a little bit differently. So we can look at a scatter plot to see what kind of differences we have here. And this is just comparing the RMSD change from the input compared to the energy. So the energy range is about 30 Rosetta energy units, and things moved about 0.6 to 1 RMSD shift. But I really just want to rank everything by energy, so that the best energy was -907. So around 400 Rosetta energy units improvement is significant. So there was a lot of very minor shifting that needed to occur to relieve these non-ideal conditions. And the total backbone RMSD shift was around 0.8. So I just wanted to see if I can get some further improvement. So I ran another round of relax using this best structure as the input structure. So I think I ran 20 repeats on this one. So I didn't think I needed a lot of trajectories, and we just got about 4 Rosetta energy units improvement. So I just moved that final structure to its own little folder, so we can see that this is the best structure energetically, and that's a good starting point for running design. So in the event that we don't have a crystal structure, we also have protocols for structure prediction. And the Rosetta community has developed a whole bunch of tools for structure prediction. And most effective ones are based on homolog structures. So the protocol basically takes a template, threads your sequence onto it, and then wiggles that protein to optimize the conformation given the sequence difference. But it has a lot of extra steps where we're sampling other conformations from up to 30 homologs, and some ab initio fragments are thrown in. But we have a lot of automated protocols in-house. We can do just a regular globular structure prediction tool, single chain homology modeling, or antibody homology modeling in bench. But we have a lot of automated protocols. So if you're a bench user, for an extra fee, we can do some very specific protocols, such as doing structure prediction with a ligand bound. And this can be much more effective than doing a structure prediction and then docking the ligand, because you've already got the binding mode for your ligand in the homolog template in this scenario. So when the homology modeling tool is modeling the conformation, it's going to capture those very specific ligand interactions during the structure prediction. So we can see here, there's a couple loops that were able to make the native-like interactions because a good homolog template was used. And then we also have in bench an additional tool for loop modeling, just to sample alternative conformations and those key loops that might be interacting in an active site. This tool was benchmarked with residues up to 17 amino acids long. and its accuracy in about 75% was under 2 angstrom RMSD. So it's oftentimes very useful. So in this scenario, I didn't need to do any loop modeling because the active site is a place I'm really leaving alone, and I'm identifying mutations in other parts of the protein to optimize the Rosetta score. So one key thing to keep in mind with the Rosetta score is it's scoring how energetically favorable your protein conformation is. And an enzyme is a little tricky in that scenario because an active site will oftentimes have energetically unfavorable residues so that it can go through these conformational shifts that are necessary for its activity. And that is not a solved problem in the modeling world. We don't explicitly model that conformational shifting in the protein to go through that activity because that's a high energy thing. So we can't model explicitly things that go through that activity. So the default Rosetta method is to find stabilization, and stabilization can kill an enzyme. So as I mentioned, we're stabilizing away from the active site. That's one way to avoid killing activity. And even though things away from the active site can affect activity, the trial and error is the best way to avoid that. Additional methods would be to bias your mutation sampling towards things that are found in evolutionary related enzymes. So oftentimes we'll take your sequence, create a PSSM, and then use those evolutionarily sampled mutations to see if those are compatible with this structure. And that can enrich the probability that we can find stabilization with a less probability of affecting your activity. So next, I want to go on to talk about how explicitly we can find mutations. The last talk had a really interesting example where they did the site saturation mutagenesis. We've got a great tool to do that in super high throughput. It's called DDG, or delta delta G. So we can sample site saturated mutagenesis 5 to 10,000 mutations in a single day. And this is high throughput, so its correlation is about 0.69, and Rosetta can do much better than that. But in a high throughput way, this is a great way to identify some really good potential hits. And we optimized this protocol in-house by increasing the amount of conformational sampling, using the most updated force field to get better scoring. And when we trained the weights on this protocol, we used a more representative data set because this initial protocol, which is really good, was a little more biased on certain types of mutations. So we believe that the predictive power here is more accurate than the previous protocol. And so just to run the DDG protocol, you just click on your structure and you click DDG. So then it opens up a tab here where you can tell it which regions of interest you want to look at. So it'll show you a chart with all the positions, tell you what the original residues are, and give you all the possible mutations. So you might just want to do a quick alanine scan through the whole thing. To not be super exhaustive, or you can focus on a region of the protein. So I identified one region of the protein, which is defined in this section. So I can say, just sample all possible point mutations in this region. And so it'll do that real quick. So just to show you what I'm focusing on here, here is this optimized structure. I chose one region that is a good spot away from the active site, but looks like a good space where we've got interacting residues that might be good for finding stabilized in combinations of mutations or single point mutations. So, so this region right here is what is listed in this DDG. So we can sample all these point mutations. So once this protocol is run, what it'll show you is for each position, the Rosetta energy score change basically after that point mutation has been done. And it's just a local wiggle. It's not wiggling the whole protein. So some aggressive mutations could be a little wrong, but you can see that it identifies some improvements. But since this is an enzyme, you'll get more regions that are unfavorable in their original position position, there's a lot of residues that have stabilizing mutations, especially in the active site. But combinations of mutations are more likely to get you significant stability. I mean, you can oftentimes find a point mutation that will increase the TM a couple degrees. The occasional lucky one will get you 5. But if you want like a 10-degree improvement in TM, oftentimes you'll need a combination. So in this example, I've chosen these residues here, and I want to allow the protocol to sample multiple mutations in this region. So I use this optimized structure. I click the relaxed design protocol, which basically is running the relaxed protocol, but on each iteration when it's sampling, it's picking a random position that I'm allowing it to mutate, and a random mutation of the set I allow it to sample. Creating that mutation, wiggle, score, and deciding if it wants to keep it. And it keeps that trajectory for 10,000 steps, basically, and decides if it wants to, it choose whatever the most energetically favorable combination of mutations, and it shows you the structure for that. So if I just want to allow mutations in this region, I can tell it to sample everything, or I can say, don't add any extra cysteines or avoid prolines, just to eliminate things that are reactive or likely to significantly shift the conformation. So then I oftentimes want to do like 100 repeats of the protocol so that I can have a wide variety of outcomes, and that will give me a library of things that are predicted to be favorable. So let me show you what the relaxed design protocol came up with. I ran 100 structure prediction, 100 relaxed design trajectories. So there's 100 structures that came out, and they've all got their own final energy. So if we want to look at the scatter plot here, we can see that, so the RMSD shifting was at maximum 0.4 RMSD, so not a huge movement, which is a good thing when you're dealing with an enzyme. And if you can see that the energy range was about negative 895 to negative 915. Now the input was 911, so we're hoping for things that are lower in energy. But of course, the key factor here is what sequences came out of this. So if we look at the sequence logo, then we can see what kind of library I can work with. So it's showing me every position, but I just want to look at the regions where I was allowing point mutations. So I can see that in position 14, we likes the valine and alanine. So it's a pretty focused library here. It's really chosen not a huge number of mutations that could improve that spot. I can, if I don't want to sample a huge number of possibilities, what I can do is just select the most energetically favorable sequences by clicking maybe the top 10, and then just look at the sequences that is prevalent in those most energetically favorable to have a more focused library. If I'm worried about having a lot more trial and error, I'll go ahead and select everything to try every sample. But I took the most energetically favorable sequence here and relaxed that structure to get a good idea of what the energetic effect of these designs would be. So the score came to negative 915, which, comparing that to the input structure, is negative 911. So the Rosetta energy score change is 4, which is significant, but not huge. So there's a good probability that this is a stabilizing set of mutations. Let me just superimpose these on top of each other so you can see that structurally we're not looking at a huge amount of shifts, but if I click on this button, it'll highlight the actual point mutations. And let me zoom in on that. We'll go ahead and put that in full screen mode to get a look at what kind of mutations we've got here. So the point mutations, a little bit of space filling mutations here, maybe on the edges. This might affect the solubility. But you can look at the individual mutations to get an idea of whether you believe them. So that's about my time, and it gives you a good overview of the Rosetta workflow that you can use in Bench for doing the design protocol. And I'd love to take any questions.
Hey, yeah, thanks so much. This was a really cool talk. I have one question maybe ahead of the Q&A answer window. Are you guys working on any new tools for this GUI or like, I don't know, interesting functionality that might be nice to have that's not quite ready yet? Do you have anything that you're going to add to this software?
Not to this software specifically. We develop APIs for people that have a specific need. Because there's so many specific needs in the Rosetta community, like enzyme people will create a set of APIs that are useful to them, and antibody people will create a set for them, that sort of thing. So the GUI itself has the most useful things that has the broadest use. So we're not expanding it.
Great, cool. So we do have a couple questions in the window. Ocean asked a question to the panelists, but I was moved to the Q&A window, which Azario did answer. They asked, is it possible to export the single mutation structure? Which,
yeah.
Yeah.
So if you just click that, you can download the whole PDB or just like the FASTA or the raw scores for that structure. So it gives you the Rosetta score and then the individual components of the score.
Great. So we also got a question from Margaret Winkler. I'm going to ask Margaret if she wants to answer live. I can promote you to unmute. So if you're prepared to ask, you can go ahead and ask. If not, I'll ask it for you.
Yes, thanks. Hi, Karen.
Hi.
Thanks for the nice talk. I was wondering, you said that active sites usually have so energetically unfavorable residues. So would it then be possible if you had, say, a crystal structure of an unknown protein to discover the active site residues by looking at the most unfavorable residues?
Yeah, that is definitely a workflow that, so for example, normally when you're looking at a DDG, the majority of the protein looks like this with the occasional favorable thing. But if you're scanning an enzyme, you're going to find things like this where almost everything is unfavorable. And that's going to be, that really focus you on key regions. Unfortunately, there's like a lot of them. So some of these might have to do with, I mean, you never see this kind of blue in a whole region. So, I mean, it'll over predict, but it can definitely narrow it down from like 500 residues to maybe like 50 key points. And if you look at the actual, so I actually highlighted every position that had an a highly unfavorable current residue. And you can see that, let me just put that in green. You can see that it's all over the enzyme. And so a lot of these are surface things. So that implies that there's an interaction with another protein partner in that region. So that can be one reason for it. But yeah, the key active site residues are also going to look super unfavorable. So that'll like hone you in, but it won't like definitely identify.
Thanks.
Great. So we have another question here. Can you work with metal cofactors for the protein homology optimization?
For the homology modeling, yes, definitely. If the template has metal cofactors present, so oftentimes we need to add an extra tweak when a metal is present. I say often, it's maybe a certain, maybe 10 to 15% of the time, because metal in modeling is not, like the full partial charges aren't quite as accurate in the modeling world. So they generally tend to stay in the correct position, but sometimes you have to add an extra constraint when you're modeling with a cofactor present to keep the orientation in its correct position. But in most cases, like I often model like zinc binding proteins, and that's that sort of thing, and the zinc stays in its correct orientation. And like magnesium and stuff can really help like a DNA stay in its correct orientation, even without that extra constraints, but sometimes that's needed.
Cool. And I think we have time for one more. We're almost at time. Roberto Chica asked a question. I'm going to ask him if he wants to be promoted to ask live. So allow him to unmute himself. If you want to ask, go ahead. If not, I'll ask for you.
Hi, can you hear me?
Yep.
Okay, great. Very interesting talk. Thank you. My question has to do with, you showed us the result of multiple relaxed design trajectories, and then you generated a web logo from them. So I'm wondering if you then optimize the structures for each combination of mutations, because some of them may not be good together. And if so, how do you guarantee that you found the lowest energy conformation for those combinatorial sequences?
Yeah, so you definitely have a wide combination, and it's only showing you here that valine is most common amongst it. And that alanine is certainly present. So what I'll often try to do is download the sequences of all the favorable things and find where for each sequence you'll want to see of the most energetically favorable, what are the unique combinations for that one. So you can, I'm not sure if I'm explaining that correctly. So you can download it and look and see what individual combinations work together. So you can look at just each unique sequences on a whole. And you can see whether this combination is the best by doing some additional relaxes to allow the conformational shifting to adapt to those sequences, and then look at the most energetically favorable score and compare those scores. And that kind of overfits the data though. So it's in general, it's a good library to test. But if you really want to narrow down your library, that extra relaxing can find combinations that appear most energetically favorable. Does that explain it?
Yes, thank you.